Um, Good afternoon, everyone. So I think we missed one of the class, I think, from yesterday, the basic biology, right? Basic cancer biology. So I think most of the discussion, what we're going to do, I'm going to do a very short discussion on the cancer biology because we need to cover some of the aspects of cancer biology. It's important as we started cancer biology program. So that's why I thought put put a couple of slides to tell you a little bit of the story of the cancer biology so that you all understand why that is important, how to study, what do we do. More than what you heard last 2 days, morning talks were intense, understanding, deeper, and translational and stuff. But probably what you hear now, very basic. I'm just giving you a concept or ideas of, cancer biology, uh, trying to establish that how you overall can think of, doing something in the cancer biology as a methodology or understanding it, and so that before you start working towards cancer biology. So, very basic, most important thing is in the cancer, it doesn't matter whether the normal cell biology, you all know, regulated growth and division, organized tissue architecture, coordinating cell or signaling communication, anything happens, damage. I told you about last first talk that any damage, any inflammation, our body can take care of it. So our normal cells can do anything anything happens to your body, you'll be fine. Next day or 2 days or later, after 3 days, any fever comes, the body will react and inflammation happens. It will come back, and normal cells can do whatever happens to your body, they'll take care of it. As we age, as I said, probably it's difficult because of the cells are normal cells, not normal cells anymore. They're getting aged. All our cells, like sometime they stop growing, right? Some of the organs I'm talking about. So those are normal cells, and DNA, all these things you can study yourself. It's not like, normal cells and differences between normal cell and cancer cells, regulated part. What I'm giving you a hint is that the very important hint that we learned many years that and understood what is important so that you don't have to spend a lot of time figuring it out. You all learned as in your undergrad and post graduation and maybe during the research program, everything. The most important thing is that the concept that you want to develop is what I think most important in this whole program, right? So that is what I think the key factor. You can go back and study. Nowadays is like easy to study. There are a lot of information that you can get it in just in your hand, with the phone, and you can actually search and get all the information. The information that you're getting, the concept ideas, probably you can also get that. It's nowadays is like AI models. You can actually develop all this, create all these ideas and all those things. But the experience, the reality, probably it is very difficult, right? The reality is, I think, in the way doing the research or want to conduct a research, or you're stuck with something and starting all over and figuring it out. What is the problem? How to overcome the problem? These are all the kind of the thing I think this program trying to help you more than trying to understand what it is, right? So one of the key issue in anything that you want to study nowadays and now on is that understanding the cell-cell communication, right? You should not forget there are just not one cancer cell can communicate with other cancer cells too, right? And one cell can communicate with other cell types, okay? And then one organ to another organ. So that's why it's just not the cell-cell communication. It's communication between many organs, communication between different cell types within. I think the understanding that I think is a key factor. You don't have to do the research. You don't have to, if you want to do the research, you don't have to understand the whole thing. As I explained in the morning, just I started with SARC, right? So I don't have to understand the whole, I learned the tumor microenvironment and all those things on the way while going. You don't have to know everything and start your work, but only the concept ideas you should know. But when I started SARC, I knew the tumor microenvironment. But if I start thinking about tumor microenvironment when I'm starting SARC, probably I would have not able to achieve because I'll get lost in the ocean probably of science, right? So you need to know all those things when you're designing your experiments and want to conduct experiments or want to start somewhere. You need to know all the cell-cell communication happens and cell communicate through what type of receptors and also signal through where. And also you need to understand that, okay, for example, say SARC got activated. Remember, end of the day, what I told you is that it is through amphiregulin, EGFR, and SARC get activated. EGFR can also activate STAT3 and the MEK and all those things, right? That is what I'm trying to tell you. So each cell, right, each cell have a lot of receptors and they communicate with many signaling pathways from one cell to another cell. There are a lot of extracellular components you need to understand. But you need to know at least before you start working. But you have to get there though. You have to go there. But you cannot do all the experiments at one time. Yeah, it will take time, but you start figuring it out one by one. But I think you need to know before you start any of those signaling. You should be aware of that this is how the cells communicate, right? I think that's what I think overall I'm trying to give you a concept. You don't have to understand what is this signaling pathway, what is this, no. It's just I'm giving you overall that you need to know, understand, and those cell-cell communicate, okay? Once you understand the cell-cell communication and you know that there are receptors and there are pro-ligands and activate and all those things. So in each signaling pathway, there are different cellular receptors and can activate through ligands and different ligands. There are many ligands. There are many cytokines also activate. This kind of thing, I don't want to bore you with many of those simple. There are many cytokines, chemokines activate many signaling pathway. I don't want to bore you all these things, but I think the concept ideas I'm trying to give you is that they can communicate. You should know that you're aware of all this activation or everything happens through some signaling pathway. Whenever you're trying to establish some research and idea, so you need to know that these are all happening like this, okay? And then because cell communicate, we all know now. So, but how do you study, right? How do you study all these cell communication? If you are interested and if you want to know, it's like in our study went into a STAT3, MEK, and also PD1, anti-PD1. So you cannot just say, I'm going to do the immunotherapy. No, the question is why you're doing immunotherapy. Then you have to understand those cell types before you do the immunotherapy. So, before you conduct anything related to immunotherapy, or you want to target something cytokines are released in the tumor microenvironment or extracellular, right? If you want to block one of the cytokine, then I will ask you, why are you blocking this cytokine? Tell me the story, and then you block it, right? As a researcher, trying to understand the biology of it, you need to know that this cytokine can be blocked. It's important, but you have to explain to me why you are blocking it. So when I did the immunotherapy, before that we screened everything, all the cell types. Then we selected these cell types are important. We need to block this. Then the PD1 was very high. That's why we did the immunotherapy. Like that, there are many research we have conducted for many targets and for many reasons, right? So I'm not showing you all this. Only I'll give you one example, gave you one example today just to tell you that this cell-cell communication is important to study. When you fail, when you are doing the cell intrinsic study, means that when we failed in the tumor intrinsic study, we were trying to target the STAT3 and MEK, and we failed. We did not really improve anything. Then comes the tumor microenvironment. You should not just go, hey, I want to study T cells. No. Tell me why. Why you want to study, right? We failed and we studied, and you need to know that why you want to study. T cells. People do many things. It's important that people just start with the T cells and start with the B cells and some macrophages and neutrophils. The people say, I'm studying a neutrophil. They have no idea why they're studying neutrophils. They give some random ideas, but probably I may not buy it. I may just say, oh, great, awesome. Okay, go ahead. But I may not buy it. But I just say, awesome, great. But oh yeah, neutrophils are important. But our story, that you build your own story to study one of these cell types, right? Your story should be convincing to others, convincing to other researchers or biologists, and tell them the story that why you want to study neutrophils, right? Why you want to study T cells or the tumor intrinsic study. So in our case, I failed in tumor intrinsic study because the target that I was actually selected did not improve anything. That's why I was actually looking for a lot of other cell types. Maybe there is something else is going on in these cells. Then I went around and looked for different cell types. So here, if you see here, this is the cell-cell communication. But if you want to study tumor microenvironment, you can actually do the single-cell RNA sequencing, right? Remember one thing. If you don't have the facility, you don't have to do single-cell sequencing. So I did not do in all my work all the single-cell sequencing. Everyone who actually conduct the single-cell sequencing, they have to put their data out there in the public domain. Okay, you can actually download and you can actually see what they have done and collect all those information and do your own research. You don't have to do single-cell RNA sequencing. Important is that hire a good bioinformatician who actually understands the biology with you, right? You have bioinformatician in coffee and take them for dinner, work with them and together. They will actually give you awesome science to you, but you have to give them the idea though. They have no idea about some biology. You give them the biology. You need to know single-cell sequencing. You need to know how to download all these public domain single-cell sequencing data sets in each and every cell type that you think of. It's already out there. You don't have to do research. But in our research, when we 50% of the single-cell sequencing, we download others' work and we cite them, and also we compare with our single-cell sequencing data, the difference between them, and also there may be a variation. It's okay. But important is you learn your data, what you have done, and you know the reality of your data, and also you know what you are trying to explain to others, whether it is true or not, right? So another way of studying the different cell types are spatial proteomics, spatial transcriptomics. The spatial proteomics are these that, if you see in this picture, they're all there, but I just wanted to give you an idea, is that there is a tissue and there is a protein expression. Yesterday I presented that one of the spatial proteomics data where I did the pan-CK and also another marker, a macrophage marker, and also we selected that area after the staining the tissue, selected that area and did the proteomics and studied. And there are a lot of data you get from that study, but already those who are published, already out there. You don't have to do it. Even so, spatial proteomics, spatial genomics and transcriptomics, what you can do, you can select that area and do the transcriptome of that study. For example, you select the T cell area, you stain the T cell, stain the neutrophil, stain your tumor cell, and you can select that area and do the transcriptome, genome, and then see what genes are changing here and all those. You think about something. And this can be easily done by the bioinformaticians if you give them the knowledge. It is a give and take business, right? You have to give them something, and they want the paper, you publish with them, and that's how we develop the many programs in the scientific publication. So, as I said, if you don't want to do laboratory work, at least you have someone, bioinformatician with you, who can actually work with you. You can publish a lot of papers. You don't have to, because idea is yours. You have to come up with idea. For example, I published single-cell sequencing data out there, and I move on. I will not use all this data in my research at all. I can only use one data, and I only select one, I just move on, right? And there are tons of data right there, and that is for the public. That's for you. If you want, if you have an idea, if you want to do something, you don't have to work in the laboratory. You can actually think of how to, get this data and analyze and do something, right? So I'm just giving you a hint so that you know, when you say, I don't have the facility here, I don't have that, I actually sitting here and I'm thinking, okay, I know that. I know there is a facility. I know there is a problem. I know there is a problem. But you know what? You're a scientist. You should be able to figure it out how you can actually develop. But I was giving you one story to Joshi is that even though when I worked in DFRL, right, there was no facility. I did finish my PhD. I was the first PhD DRDO scholar out of defense laboratory, the first one. And then a lot of PhDs came out. The reason I will tell you, it's important. If I say I don't have this, I don't have that, I would have not able to finish my PhD. I would have I worked in, I did my PhD thesis work in CFTRA, University of Mysore Biochemistry Department, NIMANS, and IASC, and I went all around to get the data. So it's important that wherever you're sitting, probably you will not be doing it. It's just a bench space. For me, the laboratory was a bench space. That's all. I just go there. That's it. Sometimes I just go there, sign, and then go out and do my work, research work somewhere else. That's how I have developed my research. I don't go to the laboratory just to do my lab. Probably I will not get anything there. Okay, it's just a space. It's just a Mangalore University, probably just a tagline for you. But you may be working somewhere else. You getting the data from somewhere else. Maybe you go to Indian Institute of Science, or IISER, or Pune, or Chennai, or anywhere you go to Hyderabad. But if you say to me that I will not be able to, I don't agree, because that means that you will not be able to. Okay, you are not able to do it. That's why that you're saying that you will not be able to because the facilities are not there. No facilities. If you think about the facility, even University of Miami facility and everything, that no, there is lacking. Because there is something that if I want to do, it is not that all the data setup came out from the university where I am. No, it is not like that. There are a lot of stories that I can tell you. If you want to do it, you can do it. You will do it. It's just that we have to decide whether you want to do it or not. So here, I think Joshi explained a little bit here. I just don't want to go in detail of the cancer. I think you already know hallmarks of cancer. Overall, the idea of signaling pathways, different signaling, invasion, metastasis, all these signaling pathways, cell death, and all those. You cannot do everything. Remember, I was not doing anything, any of those things. You find out there may be people published the hallmark of cancer, but you know what? Just for the review paper, if you want to write. If you want to do research, find out your own way. Doesn't matter where you start. Doesn't matter where you start. You always do something, literature survey, and start somewhere, right? So these are all the hallmarks for understanding the idea, concept, and these kind of things. But when you want to do it, that is a different story, okay? You don't have to worry about what they have published. But important is that understand what is out there as in cancer and before you start something to work on. Here, when you say that transformation from normal cell to a cancer cell, it's a normal cell. It is not the overnight it will become a cancer cell. No, it takes many months, years to become a cancer cell. Right? It's just not. It damages your cell, and it will normal cell become a normal cell again. It will damage and then go back. But there is something happens. For example, you smoke. Some people get lung cancer. Some smoke and they got some other cancer, breast cancer. Some got different cancer, maybe brain, or some pancreatic or colon, infected colon, or prostate, and many things. Smoking can affect many organs, but it's not just the lung cancer. It all depends on the human body, and we don't know who gets what type of cancer, right? It's hard to say. And also, in your body, so we don't know which organ is really weak. It's hard to say. Or which organ is not really not able to recover back. That's why you get the damage again and again and again, and this will become inflammation and other things, and then it will become a mutation change, genetic makeup changes, and that's why you're getting the cancer in in different organs. It's not lung, things like that. So when normal cell to become a cancer cell takes years, right? So it's a consistent damage and also change in the genetic makeup. So it takes a long time. So there is a metabolic reprogramming during that time. There is control of the cell, apoptosis cells. That means the cells are not dying. It will become something else, right? I also talked about tumor environment, the environment, toxic environment, or the what do you say, glutamine, sugar, and all those things. These are all affects the cancer cells. So in the cellular transformation, there are three things I wanted to tell you, very important in all these years, like so many years of that cellular transformation that people have studied, scientists have investigated. But recent years, in the last 5 years or 10 years, a huge change in the whole technology, huge change in the whole thing. That's why I said laboratory work, it's not anymore like laboratory work. Now you can sit at home and actually investigate a lot of other things that if you don't want to do it. The reason I will tell you why, because this is the ICGC, it's called International Cancer Genome Consortium, and there is a TCGA, that is a Cancer Genome Atlas, and also PCAWG, that means that pan-cancer analysis of a whole genome. HTAN, there is a recent HTAN data that is called Human Tumor Atlas Network. Okay, you can get any patient data you want to, any clinical data you want to. It's all available, public domain. Okay, it's all available. It is not that because that when you say there is a problem here, that means that awareness is a big problem. So if you're not aware of it, that means that you're not reading papers, you're not studying paper, you're not collecting the paper information, or you're lacking in the information. That's why you're lagging. Okay, it's important when you, as a researcher, if you want to do a PhD, if you want to do a research, if you want to do something, it's not just the cancer. It doesn't matter what area you want to go into, but it matters is that there is awareness that is lacking. So you cannot blame someone because it is you, end of the day. You cannot blame your mentor. It's you. You want to do it. Why you blame your mentor, right? It's the same concept. Don't blame others. Just you're not able to do it. The knowledge, if you want to do it, you will do it. It's just that you don't want to do it, you blame others. It's like that. So just do not. You want to do it, there is availability. Create that awareness, study, read, develop your scientific awareness, scientific mind, so you will get it. So if someone else can get it, you have the same brain, you can get it too. Okay, we all have the same brain. It's just that awareness is a key thing. It doesn't matter which area, okay, it's awareness. Even you want to shop and get a sari or shirt, pant, anything like that, it's all about awareness. Where you get this, how, where, and search, talk, discuss. I think you will find out the best suit probably somewhere. It's all about awareness. It's the same thing here, okay? It's about that. So when it comes to a tumor microenvironment, I think I talked about tumor microenvironment, and I'm actually trying to tell you is that different cell type. There are tons of cell types here, okay? It's hard to study all the cell types, okay? For example, if you go to European or some other country, developed countries, what they do is they take simple one cell types and start studying, investigating something. It's fine because they are discovering something unique way. But it's about you though, what you want to study, how you want to study. As I said already in the morning, that it's important that what is that you're trying to establish in your life, in this area, is a key factor. Where you want to see for the next 5 years, or 10 years, or 15 years as a biologist or cancer scientist, and so that what do you want to do? It's all about that. You, in that direction, you have to make that roadmap. Then only I think you can reach. If you don't have the roadmap, you will be lost. So you will do different things, and end of the day, you'll lose a lot of those years. We all did, one or the other way. But when the focus is there, the focus is an important thing, right? With the focus, there is a struggle, right? You have to struggle, just not focus is not enough, right? You have to struggle too. So this interaction transformed cell and microenvironmental components. So when we talk about cancer cells, right? So it is like I was actually giving out here normal when imagine a well-organized city where every citizen follows laws and common good things. Means that is normal cell, right? We follow the rules and regulations. There are people, they follow the rules and regulations. They wash hands and they clean, and then they don't litter or anything. They just follow the rules. The road sign is a sign. This is a normal cell. That's what they do, right? But not all the citizens are same. We all have a dirty hand sometimes. We all have a dirty mind. And sometimes we go wrong ways. And we end up in a lot of problems. And we end up in doing a lot of different things and which are not right. So these are something like I was trying to tell you that cancer cells are those that they want to live better than anybody, normal cell. And they want to live, that's why they crash everything. They try to get everything from the normal people and trying to establish the best out of there. And they always will be moving. They always will be like, trying to establish something. That's just a neighborhood problem. It's just that what I'm trying to tell you is that cancer cells are different. They are they just kill everything and they want to, without any organelles, probably they can live. It's like that. So I think sometimes it's hard to study, very hard to study. It all depends on how you're, you need to understand the cell, how they are behaving. So one thing I want to say is that observation. I actually talked about observation yesterday. So you have to observe the cells also, how they're dividing, how fast they're dividing, and how they are looking, how their nucleus is. They are different. They're not normal, right? They are different. But I think observation is the key thing. Okay. So I can talk about the observation thing in many ways. Like even in animal, observation is a key thing. For example, you treat a patient with the same drug. There are a couple of different patients, but you observe them. They are different. Each one is different. They react differently. The toxicity is different, and things like that. That's why we have statistics. That we analyze all this together and say, okay, and we will say, common factor. We take it out, common factor, and we say this is how it is happening, right? So I think observation is a key thing. It doesn't matter where we go, right? In our own life also, we observe and then we determine that this is right and this is not right, right? The same way you observe. And there are molecular drivers of cancer. We talked about some oncogenes and P53. We talked about tumor suppressor P53. We talked about KRAS, things like that. The oncogenes like RAS, MYC, tumor suppressor genes like P53, RB1. There are many. So when we talk about molecular drivers, I already touched base on that different tumors have a different molecular, oncogenes and tumor suppressor genes. So you need to know that all those things, and also you need to know that what type of signalings they have in each cancer. You need to understand the signaling pathway. They're all different. Okay. Then you have to study those signaling pathways. So the dysregulation of the cell cycle and up to cell death, okay? It doesn't matter what signaling pathway you're studying, you have to understand that cell cycle and the cell death pathways in each and every cell types. You need to know what is happening to the cell death and cell cycle. But nowadays is so much information available. People have done already, so much of work has been done, and it's the data is out there, like public data. I think you need to know where to find. Okay, it's about the literature survey, but it's very interesting. The science is always about if you can understand the biology, but you should not have the fear to understand the biology. Understand it. It's actually beautiful to know each and every organ how they work. Like for example, mitochondria is a main organ because it's the energy source. So the cancer cells need energy. So studying mitochondria is a key factor. So if you can normalize the mitochondria, because the mitochondria will be releasing a lot of ATPs in cancer cells. So if you can understand the mitochondria, make it normal, it's a great thing in cancer cells. You can actually stop probably how the cancer cells are dividing so fast. You can actually stop them. But there's a lot of research going on. But that's more than enough. If you can understand the mitochondria, you can actually do for your life the whole research. It's about that. So try to find what is important to study, so that you can study those things which are important. So for any cancer, it doesn't matter. So there is a cancer therapeutic approach targeting apoptosis pathway. It is a huge research there. You don't have to do any other signaling pathway. If you like cell death, that itself is huge. All you do in your life is just doing the cell death assay. All do you do the apoptosis assay. But you take different drugs, just study the apoptosis. That itself is huge. But if you can kill the tumor cells, not the normal cells, that itself is like in a huge research. You don't have to do anything else. But try to understand the mechanism even in the apoptotic pathway, and just study. There are a lot of things to understand. Just one signaling pathway for your life, right? Understand the apoptosis and how it is happening and what it is happening. Understanding that the whole signaling pathway is a great thing. So there's a nice review article there, Nature review on the clinical oncology, that actually give you a lot of information about it. Okay. When it comes to tumor microenvironment and metastasis, remember I talked about each cell type, different cell types, right? Each cell types have a role, just not the progression, even to the metastasis, right? In your cells, like in tumor cells, can want to metastasize to some other cell, organ. They have a different cell types also have a role, right? So I will come to that. That story I'm not giving you, like the way I've given you in the morning. The morning what I gave you story is different. That is for the therapeutic purpose. So the story about the metastasis is different, right? It is exactly the same way, but all the basics are same, right? The understanding the different cell types, how they communicate, and you will know one day that what cells are actually having a big role on the metastasis. You can control those cells and metastasis will be controlled, and those cells are not going to go and metastasize. And also when cells get metastasized, the reason is that cells have can change the shape. The cancer cells can change the shape and go into the bloodstream and then go to the other organ. If you can change the shape of the cancer cell, how do you want to change? How do you change the shape of the cancer cell? You have to, that itself is a huge research, right? Just change the shape. All your life, just change the shape. Make it normal. Change it back. How do you do it? It's just research. That's all. All you sit and think about it every day and night and figure it out and how they change the shape. Like for example, you put a cell culture and I just say observe. I always say my PhD students and the postdoc, observe. Observe those cells, whether they're spindle or they're like in cuboidal or what type of cells and how they are changing. And can you do some microscopy study and see what's happening? It's a key thing. If you just observe whole life, you can observe and give a thought process for the cells, how they are changing. Itself is a research. But you have to have that, you have to spend time to compare with a normal cell to a tumor cell, or how they're changing, what is the shape, why they're changing the shape, and why it is happening like that. That itself is a huge thing. But important is that, once you understand the biology of it, you can do the therapy, right? If you don't understand the biology, you will not be able to do therapeutic. So you have to understand the biology. For that, you have to have the observational skills in your mindset. That means that only comes with the reading, right? Reading, understanding, and knowing what's going on in those area. So in a cancer is when they are abnormal, like how they communicate to each other, right? So when cell-cell communication in the normal cell is different than the cancer cell, the communication. You have to understand, if you want to understand the cancer cell, how they communicate, you have to understand in the normal cell, how the normal cells communicate normally. If you don't understand normally what happens, you will not be able to figure it out. Only you always will be studying the tumor cells, and you won't know what is normal cells really. So if you know normal cells, how they behave and what is the difference between them in the signaling and how they communicate in normal cells and how they communicate in tumor cells, then only you can actually figure it out. How do I convert this tumor cells communication towards normal cells. These are the changes. You figure it out, like, how the crime scene, the policeman puts out all the stickers and all the same way. Figure it out, put those stickers, and just see where you can actually change it back to normal and start working on towards that point. Just giving you a hint that important to understand the normal cell communication before you think about the cancer cell communication, right? So what I'm trying to tell you is that awareness of, what you want to do, because you have to spend time and figure it out before you do anything. It is not just the cancer cell, right? It's just any cells you want to do the disease study, or any disease that you want to study. You have to understand what in that organ, what happens normally. Normally how that organ works. In the kidney, if you want to study the kidney, you have to understand kidney, how they function, what exactly normally happens, and during the development of the kidney, what happens, and all those things. Then only I think you will understand, okay, this is the problem. I think for this problem, to convert back to this, and what do I do? Things like that, okay? Simple concept, right? So in the tumor microenvironment, I think I talked about this, how any of the cell types actually influence the metastasis. The same way, how these cell types influences the tumor progression, right? They all have a role for, each have a different role, but they do it differently at different time point or different disease stage, right? In the tumor progression, I said stage, you need to, aware of different stages in each cancer, right? Means that early stage and middle stage and the later stage, because things change from early to, middle to, later. If we can convert the tumors to a pre-malignant, itself is a great thing. You don't have to make it normal completely, okay? If we can change the tumor cell in the progression towards normal, pre-malignant. So I was talking about acinar ductal metaplasia in a pancreatic cancer. So I want to change the Panin 3 stage to a ADM. That itself is a huge research. I just want you not only use research. If you can change that, that means that you can actually, make your patient live longer. At least they will have a cancer, but it is not aggressive. You don't have to worry. So these cancers, pre-malignant is enough. It's okay, fine. They will live for life with the pre-malignant malignancy. With just the Panin, we all have Panin in our body, but in a pancreas. But it's just that they're not converting into a cancer because there is no damage. You're not causing any damage to your pancreas, or you're not causing external another damage or hit or anything like that, or inflammation. If you have any other inflammation, drinking alcohol, smoking a lot, and things like that, or your diet changes, and probably you're eating a lot of oily food or things like that, this can change too. Any organ, doesn't matter, pancreas, can change. So that is a second hit that you're giving to your organ, right? So understanding the normal pre-malignancy and the tumor, and how these change, what are the changes, how this is changing, all this biology, if you can understand, you can figure it out what is happening, right? So metastasis itself is a journey, right? Beyond tumor. So when you say metastasis, right, there is no progression. It's already done. Its tumor is there. Just if you can control the metastasis is huge research, right? When in the breast cancer or the lung cancer, when you go to doctor, oh, it's already metastasis, I cannot help you. Even though I treat, I don't know how many days you're going to survive or how many months. It's very hard to say, right? But because the drug that they are treating is for the breast cancer or lung cancer or prostate cancer, they know all these drugs, but for the metastasis itself, oh, it's already metastasized to the different organ, then they have to find where the cells are, in lung, liver, or brain, or anywhere else. So if they want to treat those cells, they have to understand this, the other organ, and if I treat with this drug, what will happen to other organs and stuff like that, a lot of different things. So metastasis research itself is a huge thing. You cannot do everything. You cannot do progression. Progression is different, okay? So metastasis, when it comes to metastasis, targeting metastasis, when the metastasis is going on because these patients are not living longer, so you have to control. Means that research itself is a different research, right? Or by understanding the biology of metastatic cells is a different thing. Oh, the researchers say, okay, I'm doing all this progression, metastasis, all my lab work on this. Yes, it's a great thing, but you know what? You cannot do everything. So you have to focus on what is that. If you want to establish something, you have to focus on something that, like, what is important, right? If you want to do metastasis, that itself is a great thing. Understanding is a huge biology there, okay? Different organs, different tumor have a different type of metastasis. So understanding that whole thing is a huge thing. So determinants of metastasis, like you determine what is causing to these cells to metastasize, right? That itself is huge, right? There are, like, extracellular matrix. There are different organs, different proteins in the extracellular matrix itself is like, each protein have a different role and things like that. There are a lot of things that you can work on metastasis, right? And also EMT, it is epithelial to mesenchymal transition. The cell shape changes, right? I was talking about the shape changing in the shape itself is a huge thing. If you can understand how this cell shape is changing, right? How you can, if you can reverse it back, right? If you can reverse it to at least half of it, remember, these patients live longer, right? Reverse it a little bit, okay? That little bit reversal, right? What I'm saying is itself is a huge thing. So that's called epithelial to mesenchymal transition. Once the cancer cells, want to metastasize or become invasive, right? They are going to circulate in the bloodstream, go to different organs, and if you can understand how these tumor cells are going, how these tumor cells are hitting which organ to go, if you can understand how they are communicating in the blood, and in the blood what are all will be there, cytokines, chemokines, there are a lot of other factors. If you can understand what is there in the blood, just blood itself is a huge research. Remember, the cells are going, and there is bloodstream, blood flow, and it is flowing through, and there are many things can go in and out, go out, and there are cytokines can attach, and there are signaling around the cancer cells, and these cytokines can attach. And if you can understand which one to control in the bloodstream, so if the metastasis is happening, is huge research. What I'm trying to tell you is that awareness, right? Think what is going on. If you can think what is going on, figure it out with the doctor, see what is happened to these patients, and how many patients you had, and what did you collect from these patients, and if you can figure it out something your own way, just taking the blood sample itself from these patients is huge research you can do. But if you can understand what you want to do, right? Just in the blood sample, you can actually study a lot, right? So if you want to do just the metastasis, if you can understand the blood itself, blood and the metastasis, just imagine, you don't forget about cancer at all. Don't worry, cancer cells, studying, that, you don't have to worry. You can understand. So what I'm saying is that important is that understanding what is happening to the cells and where they are going, how they are hitting, and what technology you should use to analyze these things, or whether you should do the simple assay, or you should do something different technology to understand these things, and scanning or anything like that, okay? When it comes to metastasis, right, so clinical observations suggest that most cancers metastasize to specific target organs, right? As I explained, the process is called metastatic organotropism. The metastasis is the final frontier in cancer, right? For which there are a lot of therapies are needed, okay? So believe me, the metastasis is a huge thing. Doctor will tell you that we cannot treat this patient because it's metastasized. It's like beyond, okay? If we can research and find out something, anything small thing is huge in metastasis area. So that's why therapies are needed in the metastasis area, and definitely there are a lot of research they needed. It's important study as well. There are effective treatment right now, understanding how this is happening in the whole world. They are all working on it. So explore and understand the whole body, and see where the cells are going in there may be a different cell type and how they are flowing in the men and the women. If we can understand that men and women, there are many hormonal changes in men and women. If you can understand those, how they are hitting and in women and compared to men and things like that, there are many things that you can actually work on in that area too. So there is a integrative insights between the cell types. There is a linking microenvironment progression. I think I covered all those things. There are like, a lot of therapeutics they are working on each cell type, so they're trying to figure it out, where to target which cell types and all those things. So I think the important is that interdisciplinary approach in cancer biology, right? If you want to study any of this, right, without doctor, knowing you one of the clinician, knowing a bioinformatician, knowing a chemist who actually can design the drug and target the drug, and I'll tell you what to do, and knowing some technologies, like I was talking about, one of the imaging technology, I was talking about, mass spec analysis, and then I'm not covering a lot of other technologies, but what I'm saying is that knowing all those technologies, who is doing and where. Like I was telling you, during my PhD program, I went to NIMANS just to do the electron microscopy, because they are the best. So it's like that. All my thesis, I have electron microscopy, even though I was not able to even think about my electron microscopy when during my time. So what I'm saying is that because electron microscopy was important for my study, because I was doing the actin and filamin and nebulin and all these proteins, and there are structural proteins, and I had to really see in my eye that what is happening to the structural protein. It doesn't matter what I do, other analysis, if I don't see it. If I want to see it, I have to use the electron microscopy. There is no other way. So that's why I went to electron microscopy and study. But the same way that you had to figure it out, the interdisciplinary approach, you have to give them to the research. Just yourself. You can do PhD, you can submit thesis, but you know what? So if you want to do research, you have to, if you want to do hardcore research, you have to, do the interdisciplinary approach. And also the technology, the CRISPR, single cell, I'm talking about imaging and all those things, right? I know you may say that there is no way I can do it here. So, right? It's true. But remember, there is an imaging institute you can actually find in your own area somewhere. But if you go and talk to them, there is always a way. You can figure it out. They will tell you some guidance. They will tell you what can be done, because they are the expert. Doesn't matter what image you're talking about. You go to some other hospital. I saw like in a couple of hospitals around here on the road. Definitely they are doing some imaging, right? If you are interested, definitely there is a way. And they are the experts. They are from somewhere probably. But it's about the communication. It's about developing the approach, different if you want to do it, you will do it. That's all I can tell you. It doesn't matter what. Oh, you may say, oh, I don't know CRISPR, I have to find out. Let me tell you, CRISPR is just a technology. Anybody can do it. You actually figure it out. One postdoc or one graduate student or one PhD student only do the CRISPR. Nothing else in their life. Believe me, you can actually do great things in your laboratory. But you have to send them to learn some other institute if you want to do it. There is a lot of things that you have to figure it out. What I'm saying is that if you want to do it, you can do it. So the message that you need to take home is that the understanding the tumor dynamics. So understanding this, the whole tumor, how they work, what happens is a key factor. Knowledge, awareness, okay? Then only you can actually find the novel therapy. If you don't have that awareness, thinking about what is going on around, it doesn't matter whether it's a cancer biology or some other biology or any disease that you think about, it's the same thing, same concept, same design. Think about it, understand the biology before you treat. Don't go ahead and treat, right? Understand it, take time. If you want to do PhD, don't worry, if you do not have to finish in 3 years, but figure out something that will be for your life, that you can actually think back, oh, I've done great job during my PhD. Doesn't matter what. Mentor is just a person, okay? He's only there to guide you or may not be guiding you. But you're a scientist already when you decide to become a PhD person. You're already a scientist. So that's why you want to do PhD, right? You're already scientist. You have to think like scientists and conduct your experiments mentor is not going to help you, and he has or she has a lot of other things to work on. But you want to do PhD, you want to become a scientist, you're already scientist when you sign that I want to do PhD. Okay? So think like a scientist, act like a scientist, and do like a scientist. Read it here every day. See, science, when I left the country, and I only left leaving the job to do science, do research, right? So in my mind, it doesn't matter where I am, okay, I'm helping humanity. I'm helping someone, I'm helping some patient, doesn't matter. With the research I publish, it is for everywhere. It's the work, anybody can read. It doesn't matter which country I'm working in. The environment, the Lewis Pasteur, that idea of humanity and illuminating the world, right? I think that favorite sentence for me is like knowledge belongs to humanity. We actually give them something that will go to everyone. So that's how you need to think. It is just not, okay, if somebody says that, oh, you left the country and I don't know, and like you're never thinking back to coming back and all this thing, it doesn't matter. I always think about my country. I always think about my mom every day, every morning to evening. But important is that, what is that, why did we go, and what I'm doing, what I'm figuring it out, I think the key factor. But whether you're here or Delhi or in Chennai, it doesn't matter where you're doing. Important is what you're doing to the humanity, what you're doing that actually helping anyone. Doesn't matter what, doesn't matter who, right? I think, and also important, I think the bold letter here. Observe, question, experiment. This is how the future is for the science. That's how you build the science, right? It's just not the experiment. So observation, questioning, conducting your experiment, again observation. There are a lot of frustration. Definitely everybody is going through, right? It's like that. And I think in every cell lies the story of life, right? A story that, when understood, can transform the future of medicine. So each cell, if you can understand the cell, that means you're understanding the life, right? So one thing that I always tell my postdoc or my graduate students is that when you're entering the cell culture, think like you're putting the lab coat, white coat, and you have to. I'll tell you why. Because you're a doctor. You're going to a patient. You're going to see patient, that is your cells. They're from the patient. So you're putting in a cell culture, like it's a cell culture hood. You may say, I don't have the light, that means that you're killing the patient. So you're not doing right science. So you have to do right. You have to think like you're going to the clinic and you're conducting an operation on some patient. And that's how these your cells are. Think like that. So if you're contaminating that, you're contaminating your patient. And think what will happen to the patient. That's exactly how you need to think. You have to put alcohol. You have to sterilize yourself. You have to sterilize your cell culture, because these patients are sterilized. They are in the really good environment when they are operating these patients. Right? Exactly same thing. What if something cells comes on the cells? You're actually researching that is not right. You're actually killing these cells in a different way. Your drug is not killing, something else is killing. So you have to think like you're a doctor. You have to think like but that's how you have to think. Okay, doctor may say, I am the one actually saving the patient. No, you are the one actually saving the patient. Because if you kill the patient here in the cell culture, they're going to kill the patient even more. Just imagine, one research can kill so many patients. He can kill one patient, but you killing the many by doing the wrong science, right? So think logically. When you are entering a cell culture, you are a doctor. In your mind, you have to, that's important. I think Joshi knows. I always told Joshi, cell culture is like operation theater. So you have to think like that and act accordingly. If you think in your mind, awareness, right? If you think like that's the operation room, then you automatically wear your lab coat and automatically be putting the gloves and then putting the alcohol and sterilize yourself and making sure that you're not talking too much in the cell culture. You aware, that awareness is important. I think that is what I think important more than anything else, right? You are important, and how you are seeing the world will see you the same way. So I think you need to see the world better, right? Thank you. Okay, now is it tea time now? Is it tea time? Okay. Okay, we'll have a 10 minutes break and then we'll come back and we'll have a tutorial. We'll have more discussion and we'll go back to general cancer if you want to discuss normal cancer tutorial that from the yesterday's material, the tutorial 2 material, and also today I think you have another material for the morning session. We can actually discuss if anybody has any question about basic science to a translational science, basic science to clinical trial. If you are not able to understand and anything, it's fine. It's just that about, I just want to give you a hint and ideas so that it's our program, so we have to do it. Okay? Thank you. We'll go have tea. Thank you.